much for the invitation to come and present. The brief was, can you give us an update on the GRIA side of things? Because I know in general in Europe, there's a whole load of GRIN people, maybe not quite so many GRIA people, but of course the numbers of patients are catching up from GRIA to GRIN. So it's important that we keep things moving over, ticking over on our side as well. The majority of work we've been doing is on GRIA2 disorder. And I'm going to tell you just a little overview and some little bits of data that we've been doing, looking at the different variants that cause GRIA2 disorder and certain models we've been building, and also potentially some new drugs that are on the horizon. So GRIA2 encodes GLUA2, which is the most widely expressed AMPA receptor subunit. And if you have a problem with GRIA2 disorder, this presents with intellectual disability, epilepsy, autism, abnormal muscle tone, developmental delay, and difficulties with behavior, communication, and sleep. So there's a lot of issues that come up, as you'll of course understand. GRIA2 variants which produce both gain of function or loss of function are both associated with the disease. So just because a patient is sick and they have a GRIA2 variant, you don't instinctively know which is the best therapy option, because you don't know if it's overactive or underactive. And while all of these symptoms aren't identical between loss and gain of function, they are nonetheless overlapping. And so in general, the work we've been doing at UCL has been to try and characterize the variants, so understanding the mechanisms of these variants, the different classes of loss and gain of function. So we acknowledge that loss and gain of function is a very broad term, and there's lots of subdivisions within it, but nonetheless, those umbrella terms are quite useful. We have been looking at a couple of different kinds of modeling of this, so to try and try and understand what impact a GRIA2 variant has on receptor function and what changes it might make within brain circuits. And also to look at these drugs that are in development to try and work out whether they have the expected effects on variants. So I'll talk a little bit about all of this. So a little bit of background. So the AMPA receptors and the NMDA receptors are very closely related. Broadly, they have the same structure, but there's a few differences in the way they look and the way they work. So yes, there are 4 GRIA genes, GRIA 1 to 4. 4 subunits make up an ion channel in the cell membrane, so they come together around the outside and there's a hole in the middle. And when the receptor binds glutamate, that hole opens and it allows ions to pass through, you get an electrical current. In general, the organization of the receptors is like this. You have a ligand binding domain and the transmembrane regions, which are most important for the way that the receptor acts, the way it behaves. And we have some really great three-dimensional structures now of intact AMPA receptors, and this helps us understand, kind of validate all these mechanisms that we've been looking at over the years. And so in terms of what this actually means and how they work at the synapse, they work very, very, very quickly. And so this is how a receptor opens. Glutamate here is a green circle. It has these clamshell-like ligand binding domains, and when they close around the agonist, it levers open this gate region towards the outside of the membrane and then allows the ions to pass. And so this is absolutely crucial in the way the receptors work. There's a second process at play called desensitization, and that is it helps to process the signals in subtle ways, but it's also there as a defense mechanism to make sure you don't get excessive excitation. And what happens with that is when the glutamate stays bound for a long period of time, an interface between 2 pairs of these ligand binding regions breaks. And so even though your clamshell is closed around glutamate, you no longer provide any force to these linker regions, and therefore the gate closes. And this mechanism is well established, and it's known that GRIA disorder variants disrupt these processes. So if we can look at these processes, we can understand how a GRIA variant is causing disease. Okay, so the methods we use to look at this in general is a specialized electrophysiological technique called outside-out patch clamp recording. And what that allows us to do is to take a very small piece of membrane and then activate it with glutamate very, very quickly. And we have to do it this way because these receptors get activated on a less than a millisecond timescale at the synapse. And unless we can completely change the solution on our receptors very, very quickly, we can't make them work properly. And so in general, it looks like this. We have a glass tool here that's got a control solution on one side, a glutamate solution on the other side, and we make it rapidly jump from one side to the other. And we see the receptors activate when glutamate hits, and then this is an example of desensitization. So the glutamate stays on for a while, the channels open and then close. And so this is the way that we're finding out details about the variants. And at the moment, we have 51 different disease-associated variants of GLUA2, which is expressed with an auxiliary subunit called a TARP. And I want to show you a little bit of just a few examples of some of the work we've been doing. And you're going to have to forgive this slightly sleep-deprived flight of fancy, but this is, it's lonely in the lab sometimes. And there are, and this is not, when I say celebrity, I don't mean this is like not being found in the Kardashians or in Bono or, you know, Harry and Meghan or anything. This is a term that I came up with myself while alone in the lab. So this is a variant that's found at a known site of importance, specifically one that historically helped us to understand AMPA receptor mechanisms. So that cartoon of ligand binding domains, we kind of knew about that before we had the full structure. And the reason we knew about that was we were able to manipulate the genetic sequence of the receptors. And so again, apologies, this is gratuitous, but I wanted to make a cartoon to represent this. So I asked ChatGPT, can you make a celebrity receptor? Maybe have it wearing sunglasses, and there we go. So that was good. But then, of course, now yeah, I'm a stickler on my cartoons. They have to look right. And so, and obviously, I'm trying to nurture ChatGPT, trying to help it get better. That's a good start. Can you please make the little blue loops at the top more like the glutamate receptor clamshell ligand binding domain? And it then even asked, would you like me to make them appear to move? Yes. And then it went, how's this? And it's just like, and then I asked, okay, thanks, but I'd really like the clamshell structure. This looks more like a GABA receptor. And ChatGPT basically got the hump. I checked the chat again this morning. It was still thinking. So if the internet has been a bit slow for you today, if your emails, I'm sorry, I think I may have broken it. So for now, we're going to go with Vince the variant over here, and let's quickly move on. So just to so we're on our Hollywood Walk of Fame of variants. Okay, so threonine 707. Now, here is a cartoon representation of a GRIN receptor. One of the subunits is GRIN, and all the places that variants have been discovered on that GRIN subunit are shown as red spheres. And if we zoom in a bit on the ligand binding domain, I'm going to talk about these three positions here. Now, this was the first one that kind of made me realize, oh, wow, you know, the researchers have been looking at different parts of the receptor and finding they're important for 30 years. And now the patients are turning up at the same position. And whereas in general, the ligand binding domain variants are towards the back end where the two dimers stick to each other, this one's kind of stuck out here. And often that can mean, well, maybe this is just a bit random variant, it's not causing disease, but maybe it could be important. And in patients, changed to methionine. Now, this paper from 2005 demonstrated that threonine 707 helps to stabilize ligand binding domains. So this is the reason why, even though it's away from the rest of them, it's still important, because these bits come and make contact with each other when glutamate binds. I won't go into why I didn't spot it straight away, but everything is shifted by 21 amino acids. When you look back in the papers, I can tell you that some of the time, because I think I'm already threatening to run far too long. Okay, and so in terms of what we can actually find out about this, we jump it into glutamate, and we also saw something quite interesting in terms of the behavior of this. So typically, even for loss of function variants, the activation of the receptor is not drastically slowed, not drastically changed. But we found with this variant, the activation was a bit slowed. Deactivation was far too quick, and it's because we've lost this interaction here. When glutamate binds, the cleft can close, but it's not stabilized, and it's just a much shorter opening, and much less stable thing. And because of all this, we're pretty confident this is a loss of function variant. Okay, but again, very different from the other loss of function variants that we found. So again, these umbrella terms have there's nuance in there. Next one, leucine 504. Again, here we go, and it's this one here. Now, this one is one of the most famous of all of the residues. Again, you have to take 21 off if you're experienced in looking at AMP receptor papers to realize. And leucine 483 has been used and is still used whenever, if CFER do any AMP receptor work, they actually introduce this variant to block desensitization. And so it's kind of it's very well known and established. They don't actually stick to each other, but they kind of help to stabilize across the ligand binding domain dimer. And when the receptor desensitizes, it breaks. Now, This proline is kind of a slightly weird amino acid. It's chemically, it's a little bit, yeah, and but it causes a change in the shape of locally of the protein. And what this variant seemed to do is give us very fast desensitization and also very slow recovery from desensitization. This is another property we can look at. So the normal AMPA receptor recovers quickly. This one recovers really, really slowly. And typically, you'd think these are both loss of function properties. In actual fact, this occasionally can mask a gain of function property, whereby it holds on to glutamate for too long, so it can't recover from desensitization. So at this stage, without testing the glutamate affinity, which is a line of experiments that's easier to do in an oocyte than it is in a patch, this one is still of uncertain significance for us. So we've learned a lot about it, but we can't say for sure if it's loss or gain. And then finally, this one, E507G, which is this one here. Now, this one has been examined more recently by Derek in the front row. And this is, it's kind of just up from 504. And again, it seems to be involved in some of the same things. It seems to stabilize the ligand binding dimer in the open state. And when it's desensitized, this thing is ruptured. In terms of what we see when it becomes a glycine, this becomes a much shorter thing. This interaction is lost. And we see incredibly, incredibly fast desensitization of these receptors. So 0.2 of a millisecond. In general, the receptor is supposed to wait this long before it turns itself off. But in terms of this variant, it's incredibly, incredibly fast. And so we can be certain that this is loss of function. And so I guess all of that is to say that because we already knew they were important, it's not a surprise that variants are showing up at this place. And I kind of want to finish on this variant here, which is just next door to the one I've just talked about. But everything appears normal with the receptor properties. So it kind of just floats off upwards. It's not really doing anything in the resting state, the desensitized state. It doesn't seem to do anything there either. When we look at deactivation, it's unchanged. When we look at desensitization, it's unchanged. And I looked at this one, I thought, well, maybe, even though it's a slightly intriguing position, maybe this is not the cause of this patient's disease. And then a second patient, completely unrelated, showed up with the same variant. So this must be, the chances of that happening as a coincidence are incredibly, incredibly small. So almost certainly this amino acid here is doing something important, but just as yet, we don't know what it is. And so this kind of flips it around a little bit. So this time the patient has come first. So what can this potentially tell us about the variant? I mean, in what way is this important for AMPA receptor biology? Because the structures seem to suggest it doesn't really do that much. And it also raises the question: how broken does an AMPA receptor need to be to produce disease? So if we're on a 5 normally with our AMPA receptors, if you go to a 0, or if you have a variant that turns it all the way up to 11, then you know that you're going to be sick. But what about, if it's a 6 or a 7, is that enough to make you poorly? Or is it a 5.5 enough to make you poorly? And is it possible that even though we couldn't detect it in our experiment, the receptor is defective enough to produce the disease? And so that is something that we need to consider. And it's stuff that we kind of talked about on the first afternoon, actually, because we need to work out how we can adapt our protocols to detect whatever change this is. And we still don't really know what it is. And in first efforts to try and find out, I've done all the things I talked about on Friday. You slow yourself down, because rather than looking at homomers, now you've got to look at heteromers. Maybe it's when it's 2 GluA2s, it doesn't matter, but when it's a GluA1 and a GluA2, it does. Or maybe there's the other forms of the AMPA receptors, flip-flop splicing, RG editing, and I've looked at 3 of the 4 combinations of this, because it's the cartoon's not there, but the position of this is quite similar to the position of some of these other changes that the receptor goes through. So there's lots to learn still. And of course, it's also possible that this is nothing to do inherently with the AMPA receptor. There's something that some other protein the AMPA receptor interacts with that is affected by this particular change. And as of yet, that's not very clear. And so just as a kind of a full summary of all the GRIA2 stuff. So we've now looked at 51, the function of 51 different variants. We found 19 that we're confident are loss of function, 20 that are gain of function, 4 that have got this kind of mixed effect, some, a little bit up, a little bit down, 8 that seem to have no effect. And also there's 7 truncations and frame shifts on the database that there's no point us looking at, because they don't have an ion channel. So But the really, really key thing that we've identified is that there's a really strong correlation between gain of function variants and seizures. And that's kind of logical, but for it to be this stark is quite remarkable. So for the ones that we have identified of likely loss of function, only a single one of those patients had seizures. One of them had one seizure, so I haven't counted them. In general, of course, the trend is very, very clear. In terms of gain of function, of the 20 patients, 19 of them had seizures and one of them didn't have seizures. And in actual fact, there's a link between these two, in that both of these variants were found in the flip cassette of the alternatively spliced region. So it's possible there's something there in terms of developmental expression that allows a loss of function and a gain of function to slightly have blurred boundaries. There's various unclear ones and other ones we don't have data on. And I should say that while for the 508 patient I talked about, we weren't able to say it was gain of function, because we had this kind of data that if you have seizures, it probably is gain of function, even if we couldn't show it. We certainly didn't show it was loss of function. The clinical team decided it was worth a try with perampanel. And the last I've heard is that even that patient is doing very, very well on perampanel. So it's all kind of coming together in terms of GRIN disorder, in terms of at least identifying the best kind of treatments that we can go for. Okay, so that was all variants. Wow, okay, models and drugs are going to be quick. So why do we want to use a model? Well, once the model's established, obviously it's a very cheap and efficient way of investigating breeders. So, I forgot to tell you to look for Vince the variant. Anyway, he's there. That was that you could have earned yourself a drink. Anyway, if we can predict the functional outcome of a variant in terms of structural stuff, that will save potentially 1 to 3 months of trying to do the experiment, and then you get the answer out. So if you can jump the queue, then clearly that's for your benefit. If you can predict the effect of a variant on a synapse or a neuron on a circuit, then that saves you way more time, because you don't have to make a mouse and you don't have to do all these other really, really expensive experiments. And so it could be a way of allowing you to learn all of this kind of information without needing to do all the expensive experiments. In terms of today, I'm just going to focus on the structural stuff. And this is stuff we've been doing with Maria Oliveira from Barcelona. And this is a connection that was made at GRIN Europe a couple of years ago, and this is kind of hopefully yielding useful information already. So the transmembrane region, these kind of regions are pretty rigid. We know what they look like. It's very, very clear. One of the things I was interested in using modeling for is the linker regions, because these are very, very flexible. And in terms of the available structural data we have, it's a much lower resolution. So if you have a variant in these regions, it's a lot harder to just look at it and understand what might be going on. And these linker regions contain both loss and gain of function variants. And so if we can come up with some kind of model of how these are interacting, where they move, then maybe we can come up with a prediction of whether a variant is likely to be loss or gain of function, and potentially get onto perampanel if it's indicated. Okay. And so I must apologize to Maria, because I took like a screenshot of something you put together in PowerPoint, and I pulled it in all kinds of horrible ways. So apologies. But anyway, the model was built. I won't talk about the methods because because I will probably receive shaken heads and so on. No, I didn't do that. No. But there were three. We came up with three. Maria came up with 3 models of the different main states of the AMP receptor: resting open and desensitized. And so which variants do we want to look at? Well, we looked at a couple of variants that are just next to each other in one of the linkers. And K530M is a clear gain of function, deactivation slow, desensitization lost or reduced. And P528T, which is deactivation accelerated and desensitization accelerated. So despite the fact they're very close to each other, and the distance in general, variants in particular parts of the protein tend to do the same thing roughly in general. It was interesting to know that these kind of things did something opposite, and we had no clue as to why that could be the case. And so when we zoom in on the resting state here, we find proline 528T is here, and in the active state, it's over there. And in general, the prediction of the model is that this change to a threonine causes a weakening of one of the interactions in the open states. And so even all of these interactions around here are pretty transient. Everything's happening very, very quickly. But if you can just make something a little bit more sticky, or mean that it doesn't stick too much, that can have a dramatic effect on the behavior of the channel. And again, so the model seemed to agree for P528T. In terms of K530M, it found that the methionine that was introduced seemed to produce an extra sticky interaction in the open state that it didn't, and it didn't seem to change anything in the closed state. And so again, this is where the model very much seems to back up what the experiments have shown us. And so this is kind of really encouraging. There's some other things as well. So there were a couple of different variants found at the same site, I654T, I654V. And functionally, the I654T seems to do something. So it has, you can just about see it has this slowed activation phenotype, has accelerated deactivation, it has much accelerated desensitization, whereas the V variant didn't seem to do anything. And so in general, the modeling predicts this as well because of the nature of the chemical changes. So again, the model is supportive. And again, the in silico model seems to be coming up with useful information. However, I654V is still a patient. There's still enough wrong with this receptor that they've ended up in a neurologist's surgery. So again, we come back to that same situation. How disrupted does the function of a receptor have to be before it will produce disease? And so that's something that we're kind of continuing to work with. Okay, so enough structure, and finish on some very quickly on some drugs. So in general, as we've talked about a lot at various contexts, drugs that either directly target or counteract the active variants seem to be pretty effective treatments or they have a better than average chance of helping patients with Gris disorder. But aside from perampanel, there's no selective drugs for AMPA receptors. There are molecules that maybe at like 5 millimolar do something, but in terms of actual selective drugs that I believe are actually acting the way they're supposed to act, perampanel is the only one. So we're looking at a range of different drugs to boost or inhibit Gris variants. And just to quickly whiz through, so there are various AMPA PAMs that are in development, and various kind of other inhibitors that perampanel exists, and there's some other things that other potential. I just want to quickly finish a bit by what we've been doing on these two here. So, and so in terms of the negative allosteric modulators, AMPA receptor drugs lack specificity. So GLUA1, GLUA2, GLUA3, GLUA4, they are really close. They're very, very highly conserved. And so, especially in all the important bits, and the important bits are where you want to bind a drug to. So, unfortunately, there's no specificity between or selectivity between the different subtypes. One possibility, though, that's come up in recent years is that you can potentially target auxiliary subunits. So you have the AMPA receptors and you have what it's stuck to. And one of these proteins called TARP gamma 8 surrounds the receptor in the membrane and modifies the receptor properties. And there's a new family of drugs that have been found that bind in between these two proteins and disrupt the function. They inhibit the current. And so J&J 118 was one of the first, and that's the one we've done most of our work on, but there's also a drug called RAP 219, which is of the same family, but it's not the same molecule, which is currently in phase 2 trials, and seems to have been having a very positive outcome so far in early results. So there's certainly the chance that this could become a therapy at some point. And so with all of these experiments, I guess, it's slightly speculative, because of course, if the drug doesn't get approved, there's nothing, we can't use this information. But if it gets approved, we want to be ready to say, yes, it's suitable for these patients, maybe not so much for these patients. And so in general, what 118 does, if this is the control, you wash it on, everything gets a bit smaller, everything gets a bit flatter, and it doesn't always fully recover. Maybe, yeah, I can go into that some other time. But in terms of variance, it seems to have a really beneficial effect. So whereas the level of inhibition of the wild type receptor is relatively modest, the inhibition of the variant receptor is much stronger because you virtually lose all of this large steady state current for this variant. You're decreasing the peak current as well. You will keep the information when glutamate is released. You'll still activate your channel, but you just won't do it excessively. Okay. And so, this drug works. This one not particularly, and RAP 219 will be one of these ones, a similar effect to a similar drug. And if it gets approved, suitable for certainly some gain of function patients. And currently, there's no precision therapy for GRIA loss of function. We have loads of compounds, lots of PAMs exist, and they've been looked at for a while, but none have made it through any clinical trials. There's also not much direct evidence that they work at the synapse. We have them working in an experiment, we have them working in an animal, but the in-between steps have kind of been short-circuited. And right now, there's the most promising candidate is TAK-653, which is a PAM that's currently in phase 3 trials for major depressive disorder. And if that goes through, again, this is a drug that would be very extreme interest to GRIA loss of function patients, and of course potentially GRIN loss of function as well because of the way they interplay. And so just to say, if we have this is that 654 variant again, if you do synaptic-like applications where you're likely to have lots of high frequency activations. The control doesn't really run down much. The variant runs down much more and takes longer to recover. If you stick in this one of these compounds, it's not TAK-653, it's a related compound, then you can start to make your variant response look much more like your control than it did before. So again, these are experiments that are ongoing to try and demonstrate the usefulness of these drugs in GRIA disorder. And then very quickly, if you stick it on, you make a synaptic recording, you stick it on, everything goes a bit slower. So definitely it does work at the synaptic level as well. So I'll just very quickly gloss over that. Okay, so thank you and apologies, I'm slightly over. GRIA2 patients with seizures are nearly all gain of function and vice versa for loss of function. So Maria's built a structural model of an AMPA receptor that can predict the impact of certain of these linker-based mutants and probably many more besides. And the candidate PAM and NAM molecules can affect AMPA receptor variants and their currents. In future, we want to continue to look at variants. Loss and gain of function is very varied, but it's such an important answer to find. We need to refine the models and be ready to, if anything gets FDA approved, ready to go and know which variants are most likely to benefit. So this work is largely sponsored by the Medical Research Council and GRIN, and must acknowledge Ben Tagg and Dorota, who did various synaptic modeling and variant work in the Kilcandy Ferrant lab. Our clinical collaborator, Alan Byatt, who has a database. Any GRIA patients, please reach out to him. The more information we can gather, the better. And then, of course, Maria for her modeling work in Barcelona. Okay, thank you very much.